Lesson 3.3 is designed as a two-day experience. In day 1, the class reviews the tale of Jack and the Beanstalk, as well as any seed-related questions from earlier lesson 3.1 in the kit. And then the students will design an investigation to examine the outside of seeds. On day 2, the class dissects seeds to answer remaining seed-related questions and gather more information about the role and the function of seeds. Let's go over the materials needed for each day for lesson 3.3. For day 1, you will want to have the anchor chart from lesson 3.1 with students' recorded questions, as well as the following types of seeds from the kit. We've got cucumber, pumpkin, pea, kidney bean, and sunflower. You will want enough to give a few of each type of seed to small groups of about 4 students. You will also want to distribute hand lenses from your supplemental science kit. Finally, plastic cups partially filled with water will be needed for the end of day 1. Materials for day 2 include some soggy seeds from day 1. You'll want an Elmo or projector, hand lenses, and printed copies of the handout of seed parts, which is found in this lesson. Students should also have access to typical classroom supplies such as pencils, crayons, and glue. So, to take this lesson from the top, in day 1, students review their prior learning of plant life cycles and connect it to today's discussion about seeds. What types of seeds do students know? How would they describe the appearance of seeds? What other facts do students know about seeds, etc.? Students record what they know about seeds in their science notebooks, and the teacher can circulate and look for any additional misconceptions during this time that can either be addressed in this 2-day lesson or in the kit's following lessons. The next step is to help students identify how to design an investigation. If at all possible, we'll use any relevant student questions from lesson 3.1 as the basis to drive the investigation. If necessary, we as the teachers can introduce some scientific questions that the class can investigate, such as, what is inside a seed? How does a seed become a plant? Or, do all seeds have the same parts? Then, we'll discuss with students how they might go about investigating these seeds. What tools will they need to use? What should they be looking for? Etc. From this discussion, students should understand that they will investigate the outside and inside of seeds, using hand lenses as a tool, to see if they can answer any of the questions that were chosen for this investigation. Because students work in small groups to investigate their seeds, it's also good to take time to review those small group expectations, such as taking turns, listening respectfully, helping each other, etc. With all of that in place, the rest of day 1 consists of small groups of students using their hand lenses to observe the outside parts, similarities and differences among the variety of seeds that they have. During this investigation, the teacher is circulating and writing notes about which students observed something interesting or which students had a good sense-making comment about seeds that helped them connect the dots. After students have had time to investigate, they'll gather back as a whole group, and the teacher can call on a few students to share observations and reflect back on the investigation questions to see if any of those questions have been answered by the investigation so far. By this point, at the end of day 1, it is likely that students are able to make observations and descriptions of the outward appearance of the seeds and their physical properties, but the inside of the seeds is most likely still a mystery for students at this point. To help see the inside of the seeds, which will be the focus of Day 2's investigation, students will end Day 1 by placing some of their seeds in the cup of water and letting them soak overnight. Save the plastic cups when you are done using them for this lesson. You'll need to reuse these plastic cups for other lessons later in this kit. So now Day 2 begins. And this has students pick up where Day 1 left off by having them briefly review what they've learned so far, as well as what questions from the investigation still remain. Then, the students will investigate their soggy seeds, which they will now be able to easily split open with their fingernail or with the aid of a plastic knife to reveal the inside of the seeds. After students have had a chance to observe the inside of the seeds, the teacher leads a discussion to help students identify the parts of a seed and how each part will help the plant grow. Whenever possible, refer back to those questions that were chosen to drive this investigation. For example, a question like, "How does a seed become a plant?" is better answered in today's investigation, once we have had a chance to see the embryo and the food source contained inside the seed. It is also likely that students will generate additional questions during these investigations, and these additional questions can also be added to that anchor chart from Lesson 3.1 and kind of keep a running record of questions that come up and the wonderings of the class throughout these investigations. This process helps students see how different science practices connect to each other, such as how asking questions led to designing the investigation, which gave us evidence we needed in order to construct an explanation about how the seeds work, which in turn leads to more questions and another investigation, and so on.